My name is Jordan Kleinman. I am a third-year chemical biology graduate student in the Fujimori lab at UCSF, where I study the sequence-specific binding modality of ribosome-acting antibiotics. The ribosome is the central catalyst of protein production for the cell, coordinating a huge number of factors, including mRNA and tRNA, in order to facilitate proper amino acid orientation and peptide bond formation. As the ribosome is such a key factor, many antibiotics are known to bind to the ribosome and disrupt this reaction, thereby interfering with cellular growth. My research focuses on a class of antibiotics known as oxazolidinones, which bind at the interface where peptide bond formation occurs, the PTC or peptidyltransferase center. These molecules consist of an eponymous oxazolidinone core, the A ring in this figure, with variable ring structures surrounding, and are used clinically as a last line of defense in the treatment of drug-resistant gram-positive pathogens. Previous structures of oxazolidinones with non-translating ribosomes indicated that they acted as global inhibitors of translation, preventing any incoming tRNA from achieving peptide bond formation. It turns out that this is not the case, however, and recent studies of oxazolidinones in actively translating systems have shown that the molecules actually act as sequence-specific inhibitors of translation, causing the ribosome to stall specifically with alanine in the penultimate position. My project aims to use a structure and sequence-guided approach to understand and expand oxazolidinone context specificity. I hope to shed light on the molecular determinants involved in stalling, both for oxazolidinones and for other ribosome-binding antibiotics, and how this plays into antibiotic efficacy, as well as to create tool compounds for the targeting of proteins of interest via amino acid sequence. By improving our understanding of context-specific ribosome stalling, this project will enable the development of better antibiotics. For instance, one might imagine a series of compounds which target only essential proteins in order to destroy a pathogen while evading its mechanisms of resistance. Through application of these concepts to mammalian or mitochondrial ribosomes, we might be able to develop context-specific catalytic drugs for cancer or metabolic disease. A clear structure-activity relationship at a single site, such as context-specific inhibition at the peptidyltransferase center, would vastly decrease difficulties in drug development, especially for more canonically difficult targets. Thank you for listening to my talk today, and I look forward to seeing you at the symposium.